The overall goal of the following experiment is to use a developmentally timed manipulation of food availability to create a large range of body sizes, which can then be used to estimate the scaling relationship among morphological traits. First, eggs are collected from the same population of flies over 3 days, creating 3 age cohorts. When the oldest cohort of larvae begins to pupate, larvae of all cohorts are removed from the food. The result of this dietary modification is an enhanced range of size variation of the pupae. Next, pupal and adult wing images are taken of all individuals to allow for the estimation of the morphological scaling relationship between body and wing size. Ultimately, results are obtained that show how wing size scales with the full range of possible body size by fitting a type 2 linear regression to the data.
In this study, we use a developmentally timed manipulation of access to nutrition to experimentally increase body size variation among adult flies. This enables us to estimate morphological scaling relationships across the full range of body sizes.
This method can help answer key issues regarding the expression and evolution of morphological scaling, such as identifying the proximate basis of variation in the parameters of scaling relationships among biological groups.
Move a population of flies into a mating chamber, and then collect eggs every 24 hours for 3 days, ultimately creating 3 age cohorts. With the aid of a dissecting microscope, move batches of 50 eggs from each cohort into vials containing 10 milliliters of